Welcome to For Science Salah Lotfy channel. Today's episode takes you inside one of the most extraordinary breakthroughs in cancer research. Imagine a material, soft, flexible and rich in water, capable of reprogramming cancer cells in just 24 hours. A special double-network hydrogel that does not just hold cells, it transforms them. Researchers discovered that when tumor cells touch this hydrogel, they rapidly turn into cancer stem cells, the most dangerous, therapy-resistant cells, responsible for relapse. But here is the twist. By forcing these cells into their most aggressive form, scientists can finally identify their hidden weaknesses. This hydrogel activates powerful signaling pathways, boosts genes like SOX2, NANOG, and OCT4, and exposes new molecular targets such as PDGFR, targets that doctors can potentially attack with precision drugs. This means a future where we can detect cancer's most lethal cells early, test personalized therapies on them, and even eliminate the root cause of tumor recurrence. A new window has opened in the fight against cancer, one where materials, science, and medicine merge to rewrite what treatment can be steinsaltensites. Starts now, right here on For Science Salah Lotfy channel.
Welcome to For Science Salah Lotfy channel, where global scientific research becomes clear and accessible.
هنا نحول الأبحاث العلمية العالمية إلى معرفة مبسطة مترجمة لك بلغتك.
Ici, la science est expliquée et traduite pour tous.
Ciencia clara, accesible y disponible en varios idiomas.
Wissenschaft, verständlich und mehrsprachig für jeden.
قناة For Science Salah Lotfy channel. العلم للجميع وبجميع اللغات. تابعنا لاكتشاف المزيد.
For decades we've battled cancer with drugs and radiation, but tumors often return, driven by elusive cancer stem cells. These queen bees of the tumor resist treatment and regenerate the cancer. Now, a breakthrough, scientists can create these cells on demand using a revolutionary hydrogel. Unlike traditional methods, this hydrogel mimics the body's tissue environment, letting us observe cancer cells as they behave in real life. When tumor cells are placed on this gel, they transform, reverting to a stem-like state. This means we can finally study cancer stem cells in detail, learning how to target and defeat them. The implications are huge. We move from one-size-fits-all therapies to treatments tailored to each patient's tumor. By understanding what sustains cancer stem cells, we can develop drugs that cut the tumor off at its root. This is a paradigm shift, offering new hope and precision in cancer treatment. The journey is long, but with tools like this hydrogel, we're closer than ever to winning the war. Imagine a future where cancer is not just treated, but truly outsmarted. The dawn of that future is here. The secret of this hydrogel is its double network structure. Two intertwined polymers that create a soft, tunable environment. Its stiffness can be matched to any tissue, from brain to muscle, letting cancer cells feel at home. When tumor cells sense this authentic environment, they quickly shift into a stem-like state. Within 24 hours, many cells form spherical colonies and express stem cell markers. Previously, isolating cancer stem cells was slow and yielded few cells. Now, we can generate them in abundance, fast and reliably. The hydrogel does all the work. No genetic tricks or extra chemicals needed. It proves that a cell's physical context is as important as its DNA. This insight is transforming cancer research. The hydrogel's simplicity and power make it a game changer. It's not just a new tool, it's a new way of thinking about cancer. By recreating the tumor's true environment, we unlock its deepest secrets. Context, it turns out, is everything. The hydrogel's physical cues trigger dramatic changes inside cancer cells. A key pathway, called Hippo, is altered, releasing a protein named YAP. YAP moves into the cell's nucleus, switching on genes for stemness. This reprogramming is confirmed by the activation of other stem cell factors like SOX2 and OCT4. The result, cancer cells gain the power to regenerate and resist therapy. By mapping these pathways, we identify new drug targets. Blocking YAP or SOX2 could stop cancer stem cells from forming. This would make tumors more vulnerable to standard treatments. Understanding these molecular switches gives us a roadmap to attack cancer at its root. The hydrogel reveals not just what cancer stem cells are, but how to defeat them. It's a powerful step towards smarter, more effective therapies. Cancer stem cells are rare and hard to isolate, limiting research and drug discovery. The hydrogel solves this, letting us grow large, realistic populations of these cells. Unlike plastic dishes, the hydrogel mimics the tumor's true environment, making experiments more relevant. Now we can screen thousands of drugs to find those that target cancer stem cells specifically. We can also study why some tumors resist treatment and how to overcome that resistance. The hydrogel acts as a miniature laboratory, revealing how cancer stem cells interact with their environment. It helps us ask and answer fundamental questions about cancer's origins and survival strategies. This platform accelerates the search for new therapies. It's not just a tool, it's a window into the heart of cancer. With it, we can finally outpace cancer's cunning. The hydrogel's greatest promise is in personalized medicine. Imagine taking a patient's tumor, growing their unique cancer stem cells on the hydrogel, and testing drugs directly on them. Doctors could quickly identify the most effective treatment for each individual. This approach, called functional precision medicine, removes the guesswork from cancer therapy. It also reveals hidden vulnerabilities in each patient's cancer, opening doors to new drug combinations. Treatments become more effective and less toxic, targeting only the cells that matter. Patients gain confidence, knowing their therapy is custom designed. The hydrogel is unlocking a future where cancer care is precise, strategic, and personal. The war on cancer becomes a series of targeted victories. With this technology, hope is no longer just a word, it's a plan. Why is cancer so hard to treat? Scientists may finally have an answer, thanks to a breakthrough study published in. This research introduces a revolutionary hydrogel, a high-tech, jello-like material that can rapidly turn ordinary cancer cells into the most dangerous type, cancer stem cells. For years, studying these elusive cells was nearly impossible. Now researchers can generate and observe them in just 24 hours. Cancer stem cells are the masterminds behind relapse, resisting treatment and rebuilding tumors. With this hydrogel, scientists can finally study these cells up close, opening the door to new, more effective therapies. Imagine doctors growing a patient's own cancer stem cells to test which drugs work best, ushering in a new era of personalized medicine. This isn't science fiction. This hydrogel could change how we fight cancer forever. Science just took a giant leap forward. This discovery is the result of a powerhouse team at Hokkaido University in Japan. Led by Professor Shinya Tanaka, an expert in cancer biology, and Professor Jianping Gong, a pioneer in hydrogel technology, the team combined expertise in medicine, materials science, and molecular biology. Professor Gong's double-network hydrogel was the key innovation, providing the perfect environment for reprogramming cancer cells. Dr. Susumu Minamisawa's molecular detective work confirmed the transformation at the genetic level. Graduate students and technicians worked tirelessly behind the scenes, running experiments and collecting data. This collaboration shows what's possible when brilliant minds unite across disciplines. Together, they've changed the landscape of cancer research. Tumors aren't just a mass of identical cells, they're complex communities. At the heart are cancer stem cells, rare, resilient, and able to regenerate the entire tumor. These queen bee cells survive chemotherapy and radiation, lying dormant until they rebuild the cancer. Their scarcity has made them nearly impossible to study, stalling progress for decades. Most treatments kill the worker cells, but miss the stem cells, leading to relapse. Scientists have long known these cells are the key to a cure, but couldn't reliably produce or study them. The Japanese team set out to change that by inventing a way to create cancer stem cells on demand. Their solution could finally unlock new targeted therapies. The double network hydrogel is the hero of this story. Unlike ordinary gels, it combines toughness and flexibility, mimicking the body's soft tissues. Its secret isn't chemistry, but physics. The gel's structure physically trains cancer cells, triggering them to become stem cells. This process, called mechanotransduction, turns mechanical signals into genetic changes. No need for complex chemicals or genetic engineering, just the right surface. The result? A fast, clean, and standardized way to generate cancer stem cells in any lab. This technology democratizes cancer research, making it accessible worldwide. It's a simple tool with revolutionary impact. The future of cancer research just got a lot brighter. The experiment was stunningly simple. Researchers placed cancer cells from brain, lung, and bladder tumors onto the double network hydrogel and watched them transform within 24 hours. These new cells formed tumor spheres, a hallmark of stemness, and showed a surge in key genetic markers like SOX2. The transformation was driven by the Hippo-YAP pathway, activated by the gel's physical cues. When injected into mice, these cells were more aggressive and resistant to chemotherapy than ever before. The hydrogel had created a factory for the most dangerous cancer cells. For the first time, scientists could reliably produce and study the enemy. This breakthrough paves the way for new drugs that target cancer at its root. The fight against relapse just got a powerful new weapon. This hydrogel isn't just a lab tool, it's a game changer for patients. Doctors could soon grow a patient's own cancer stem cells, then test hundreds of drugs to find the most effective treatment. Fast, no more months of trial and error, just targeted, data-driven therapy from day one. Imagine a lung cancer patient whose tumor resists standard drugs. With this technology, doctors could discover a new effective option in weeks. This approach could save lives, reduce side effects, and finally outsmart cancer's ability to return. By unlocking the secrets of cancer stem cells, scientists are closer than ever to a cure. On November 21st, 2025, we stand at the threshold of a new era in cancer treatment. Science is bringing hope to millions.
Cancer's return after treatment is one of medicine's most painful mysteries. Patients celebrate remission only to face the disease again, often stronger and more resistant. This cycle, called cancer recurrence, has forced scientists to rethink what a tumor really is. Our treatments destroy most cancer cells, but often miss a hidden threat. Imagine chemotherapy as weed killer. It clears the garden, but leaves roots behind. Not all cancer cells are equal. Within tumors, a rare group of cells acts as the masterminds, able to survive treatment and regrow the cancer. These cells can lie dormant, evading even our most powerful therapies. Their resilience is why cancer can return, sometimes years later. Scientists now believe these elusive cells are the key to recurrence. Understanding and targeting them is crucial for a true cure. The search for answers begins with these hidden survivors. These mastermind cells have a name: cancer stem cells, or CSCs. Like normal stem cells, CSCs can self-renew and create new cancer cells, fueling tumor growth. They're rare, sometimes less than 1% of a tumor, but incredibly powerful. CSCs can travel through the body, starting new tumors in distant organs. Ordinary cancer cells can't do this. Only CSCs have the tools to spread and restart cancer. The problem? Standard treatments target fast-dividing cells, but CSCs often lie dormant. This allows them to survive, untouched, while the rest of the tumor shrinks. It's like pruning a tree, but leaving the roots to regrow. To stop recurrence, we must find and eliminate these root cells. That's the challenge facing cancer researchers today. Studying CSCs has been nearly impossible. They're too rare to isolate easily. No more hunting for a needle in a haystack. The hydrogel gives researchers a powerful tool to create and investigate the enemy. Enter a breakthrough from materials science, the double network hydrogel. This soft, tunable material mimics the environment inside the body, especially the brain. When ordinary brain cancer cells are grown on this hydrogel, they transform, gaining the self-renewing, tumor-forming abilities of CSCs. The hydrogel acts like a switch. revealing the hidden potential of regular cancer cells. Now, scientists can generate large numbers of CSCs in the lab, on demand. This opens a window into their biology, making it possible to study and target them directly. It's a game changer for cancer research. For the first time, we can study CSCs up close and search for their weaknesses. But do these lab-made CSCs behave like the real thing in living organisms? Scientists put them to the test in mice. Mice injected with thousands of ordinary cancer cells didn't develop tumours. But mice, given just 100 hydrogel-grown CSCs, developed aggressive brain tumours. This proved the hydrogel could create truly dangerous, tumour-initiating cells. The experiment showed that a cell's environment can reprogram it into a more malignant state. It's not just theory. This transformation happens in living systems. With this proof, researchers had a new way to study and attack the most dangerous cancer cells. The next step, uncovering how the hydrogel triggers this change. How does the hydrogel turn ordinary cancer cells into CSCs? Scientists found that the soft hydrogel activates a molecule called integrin beta-1. This triggers a chain reaction, switching on a protein called STAT3, crucial for cancer growth and survival. STAT3 acts as the master switch for the CSC state. With this pathway identified, researchers had a clear target. Instead of attacking all cells, they could focus on the machinery that creates CSCs. The hydrogel not only revealed the enemy, but also its secret communication lines. The next challenge, finding a drug to block this pathway and destroy CSCs. Researchers tested drugs that blocked the STAT3 pathway. One promising inhibitor selectively killed the hydrogel-created CSCs while sparing ordinary cancer cells. This was proof targeting the right pathway can eliminate the root of recurrence. Imagine using a patient's tumour sample to grow CSCs on hydrogels, then testing drugs to find the most effective treatment. This approach could personalise therapy and prevent cancer from returning. The hydrogel technology brings hope, turning hidden cancer stem cells from a mysterious threat into a visible, targetable enemy. For the first time, we have a tool to study, understand, and destroy the cells that drive recurrence. The journey from lab to clinic is just beginning, but the path is clear. With this innovation, we move closer to a future where cancer doesn't just shrink, it stays gone. The key to a cure may finally be within reach.
In the fight against cancer, a surprising breakthrough has emerged from a simple jelly-like hydrogel. This material can reprogram ordinary cancer cells into their most dangerous form, cancer stem cells, in just 24 hours. Why does this matter? Because cancer stem cells are the hidden engine behind relapse and resistance to treatment. Traditional therapies shrink tumors, but often miss these master cells, allowing cancer to return stronger. The new hydrogel lets scientists create and study these elusive cells easily, opening a new frontier in cancer research. Unlike complex genetic engineering, this method uses the physical environment, a soft biomimetic gel, to trigger the transformation. The process, called mechanotransduction, tricks cancer cells into revealing their stem-like identity. For years, isolating cancer stem cells was slow and difficult. Now labs can generate them in abundance, accelerating drug discovery. This could fast-track therapies that target the root of cancer, aiming for lasting cures instead of temporary remissions. The simplicity and power of this hydrogel may change how we study, treat, and ultimately defeat cancer. By mimicking the environment cancer stem cells thrive in, this material gives us a direct line to the disease's core. It's a game changer for oncology, offering hope for more permanent solutions. To appreciate this hydrogel's impact, we need to understand cancer stem cells. Tumors aren't just a mass of identical cells, they're a hierarchy with a few queen cells at the top, the cancer stem cells. These cells can self-renew and regenerate the entire tumor, making them the true masterminds behind cancer's persistence. Standard treatments kill the bulk of tumor cells, but often miss these slow-dividing stem cells. That's why cancer can return, sometimes more aggressively, even after successful therapy. Eliminating the visible tumor isn't enough. The root, the stem cells, must be destroyed to prevent relapse. The challenge? Cancer stem cells are rare and hard to isolate, making them difficult to study or target. Traditional lab methods fail to mimic the soft, complex environment these cells need to survive. The new hydrogel solves this by creating the right conditions to generate and study cancer stem cells from ordinary tumor cells. This breakthrough removes a major roadblock in cancer research. Now, scientists can focus on the real culprits behind recurrence. Targeting these cells could be the key to lasting cures. The hydrogel brings us closer to that goal. The magic of this discovery lies in the hydrogel's design. Hydrogels are soft, water-rich materials that can mimic the feel of human tissue. The breakthrough came from a double network hydrogel made of two interlocked polymers, hyaluronic acid and p-nipam. Hyaluronic acid is a natural component of our bodies, providing familiar biochemical signals to cells. P-nipam is a smart polymer that responds to temperature, adding a unique twist. Together, they create a soft, supportive environment that encourages cancer cells to revert to a stem-like state. No genetic engineering is needed, just the right physical and chemical cues. This double network structure is a leap forward, letting researchers control cell behavior with precision. Unlike rigid plastic dishes, this gel offers a realistic three-dimensional microenvironment. The synergy between softness and biochemical signals is what makes the transformation possible. It's a clever blend of biology and materials science. When scientists placed cancer cells from various tumors onto the hydrogel, the results were dramatic. Within 24 hours, the cells changed shape, clustering into tight spheres, a hallmark of stem cells. More importantly, their internal biology shifted, key stem cell markers surged. This rapid transformation, once thought to take weeks, happened in a single day. The hydrogel taps into a hidden potential within cancer cells, reawakening their stem-like state. Unlike older, slow methods, this approach is fast, reliable, and works across many cancer types. The versatility means researchers can study stemness in a wide range of tumors. The physical environment, not just genetics, can control cancer cell fate. This universal platform opens new doors for understanding and targeting cancer at its root. The experiment proved that the right environment can fundamentally change a cell's identity. The hydrogel's power was especially clear with glioblastoma, a deadly brain cancer. Both lab-grown and patient-derived glioblastoma cells transformed into stem-like clusters on the gel. This wasn't just a lab artifact. The effect held true for real patient samples. Now doctors could create a personalized model of a patient's cancer stem cells within a day of surgery. This paves the way for testing drugs on a patient's own cancer stem cells, moving toward truly personalized medicine. For a cancer as aggressive as glioblastoma, this is a major leap forward. The hydrogel reliably creates the environment these master cells need, validating its mechanism. Researchers now have a powerful tool to study and target the cells driving recurrence. It's a new hope for tackling one of the toughest cancers. Was the full effect due to both gel components? Scientists tested gels made with only hyaluronic acid, only p-nipam, and the full double network. The answer was clear. Only the combination triggered the rapid transformation to stem cells. Hyaluronic acid alone gave some signals but not enough. P-nipam alone provided softness but not the full effect. Only together did they create the perfect environment for reprogramming. This synergy highlights how both chemical and physical cues are essential. The double network design is what makes the hydrogel so effective. It's a masterclass in engineering the right microenvironment for cell fate. To prove these were true cancer stem cells, scientists looked at their genes and behavior. The hydrogel reprogrammed cells showed a genetic signature matching real stem cells. The transformation unfolded in a precise sequence, with key genes switching on within hours, but the ultimate test was in mice. A few reprogrammed cells formed aggressive tumors, while many original cells did not. This confirmed the hydrogel was creating bona fide cancer stem cells, not just lookalikes. The ability to start new tumors is the gold standard for identifying these master cells. The results validated the hydrogel's power to reveal and generate the true drivers of cancer. This simple material passed the toughest tests in cancer biology. It's a breakthrough for both research and future therapies. The evidence is clear: the hydrogel unlocks cancer's most dangerous cells. This hydrogel isn't just a clever material, it's a revolution for cancer research and treatment. First, it gives scientists a fast, reliable way to study cancer stem cells from any tumor. Second, it accelerates drug discovery, letting companies test thousands of compounds on the real culprits behind relapse. Third, it enables personalized medicine. Doctors can test treatments on a patient's own cancer stem cells to find the best option. Finally, it could lead to better diagnostics, helping predict and prevent recurrence. By understanding and targeting these master cells, we move closer to lasting cures. The hydrogel is more than a tool, it's a key to cancer's secrets. It promises smarter, faster, and more personal cancer care. The future of oncology may start with a simple gel. Imagine the universe within us, trillions of cells, each a tiny world, building tissues, organs, and ultimately you. These cells don't float in a void. They live in a 3D environment called the extracellular matrix, a scaffold of proteins and molecules. This matrix is their universe, providing structure, support, and vital signals. It's a constant conversation between genes and environment. What if we could build our own cellular universes? Enter biomaterials. Scientists have engineered hydrogels, networks of polymer chains, mostly water, mimicking our tissues. We can tune their stiffness and texture, making them soft like brain tissue or rigid like bone. Hydrogels have become powerful tools to study cancer, especially brain cancer, which is hard to model in the lab. Growing cancer cells on flat plastic doesn't reflect their true behavior. But with hydrogels, we can recreate the brain's soft environment and observe how cancer cells really act. A special material, the double network DN hydrogel, combines rigidity and flexibility, offering toughness and resilience. Scientists placed glioblastoma cells, the deadliest brain cancer, onto this DN hydrogel. They expected to learn about cell movement or growth. Instead, they witnessed something profound. The hydrogel wasn't just a passive stage. It actively triggered a transformation, turning ordinary cancer cells into their most dangerous primordial form. The DN hydrogel became an active participant in the drama of cancer. It revealed how the physical world can reprogram a cell's fate. This discovery opened a new chapter in understanding and potentially controlling cancer. When a glioblastoma cell lands on the DN hydrogel, a rapid transformation begins. Within 24 hours, the cell starts reverting to a more primitive, powerful state, a cancer stem cell. Cancer stem cells are the apex predators of tumors. They self-renew, resist therapy, and drive recurrence. Creating these cells on demand simply by changing their environment is a breakthrough. The DN hydrogel's stiffness sends a powerful message to the cell. Proteins on the cell surface sense and translate this physical cue into a cascade of chemical signals. The cell interprets this as a command: transform. By the 24-hour mark, the change is clear. Cells clump into spheres and express genes linked to aggressive, therapy-resistant tumors. This rapid metamorphosis, driven by the hydrogel, gives us a real-time window into cancer's most dangerous form. We can now watch and learn how a cancer cell becomes a supervillain, and in doing so, we gain clues on how to defeat it. The physical environment is a powerful lever in cancer biology. How does a physical touch become a biological command? It starts with a chain reaction inside the cell. The first domino: the cell's contact with the hydrogel activates focal adhesion kinase, or FAK. FAK is a master switch, sensing the outside world and relaying signals inward. Within hours, FAK is switched on, quickly followed by another kinase, SAK. FAK and SAK work together, amplifying the signal and sending it deep into the cell's command center. This FAK-SAK cascade is the spark that reprograms the cell. The process is precise, not random, a well-defined program triggered by the hydrogel. By mapping these early switches, we can find ways to interrupt the transformation. If we jam the mechanism here, we might keep cancer cells in a less dangerous, more treatable state. The hydrogel provides the input. The FAK-SRC pathway processes it. Understanding this cascade is key to controlling cancer's fate. The first moments matter most. The FAK-SRC signals head for the nucleus, the cell's genetic library. Their target, a gene called osteopontin, or OPN. OPN is often linked to cancer's worst behavior. On the DN hydrogel, OPN levels surge. This isn't a subtle shift, but a massive increase. Scientists silenced the OPN gene and found the transformation stopped. No cancer stem cells formed. OPN is the linchpin. Without it, the reprogramming fails. The hydrogel's physical cue, relayed by FAK and SRS, converges on this single gene. OPN acts as a master regulator, orchestrating the cell's metamorphosis. Controlling OPN could mean controlling cancer's most dangerous cells. This discovery gives us a powerful new focus. The path from touch to transformation runs through OPN. How does the signal from FAK and SRC reach the OPN gene? The answer? The sonic hedgehog pathway, or SHH. Normally active in embryonic development, SHH is reawakened in many cancers. The key player is GLI1, a protein that enters the nucleus and activates genes. On the hydrogel, GLI1 is switched on, connecting the physical cue to OPN activation. Scientists blocked GLI proteins and found the transformation halted. No OPN, no cancer stem cells. The chain is clear. Hydrogel activates FAK and SRC, which awaken SHH, which activates GLI1, which turns on OPN. Each step is essential. Break the chain and the transformation fails. This elegant pathway explains how a physical touch rewrites a cell's genetic program. The mystery is solved. Mechanics meet genetics through SHH and GLI1. Targeting this pathway could disrupt cancer's reprogramming. The story deepens. Physical forces from the hydrogel also open mechanosensitive calcium channels on the cell's surface. When stretched, these channels flood the cell with calcium ions, a flash of lightning inside. Calcium acts as a powerful signal, boosting the activity of SRC kinase. This creates a feedback loop. Force opens channels, calcium boosts SRC, and SRC strengthens the cell's grip on the hydrogel. The cycle locks the cell into transformation. Blocking these calcium channels reduces the number of cancer stem cells and dampens SRC and OPN activation. The cell needs both the physical touch and the calcium spark to fully commit to its new identity. Mechanics and chemistry work hand in hand. Disrupting either signal can slow or stop the transformation. The cell's fate is shaped by both force and flow. The hydrogel-driven transformation acts as a magnifying glass for cancer's vulnerabilities. As cells become cancer stem cells, they reveal new targets, like platelet-derived growth factor receptor, or PDGFR. PDGFR appears on the surface of these aggressive cells, making it a prime therapeutic target. Scientists treated the transformed cells with a PDGFR inhibitor and saw their stem-like abilities collapse. The hydrogel not only teaches us about cancer biology, it points to existing drugs that could target the most dangerous cells. Blocking PDGFR could help prevent cancer recurrence driven by stem cells. The hydrogel is a discovery platform, revealing cancer's hidden armor and its weak spots. It's a new way to hunt for cancer's Achilles heel. By forcing cancer to show its hand, we can design smarter therapies. The path to better treatment starts here. The impact of hydrogel technology reaches far beyond the lab. Imagine testing a patient's tumor cells on a DN hydrogel to see how aggressive they are within days. This could become a powerful diagnostic tool, guiding treatment choices. We could create a patient's cancer stem cells in the lab and test a library of drugs to find the most effective therapy. Personalized medicine becomes reality, no more guesswork. Hydrogels also speed up drug discovery, providing a rapid way to test new compounds against the toughest cancer cells. We began by exploring the inner cosmos of the cell and discovered how a physical environment can trigger profound change. The DN hydrogel revealed a hidden pathway from touch to genetic command, leading to cancer's most dangerous form. This knowledge is a roadmap for new diagnostics, personalized treatments, and next-generation therapies. We stand at the edge of a new frontier in cancer care. The cosmic dance between cell and environment is now a tool in our fight. The future of cancer treatment is being shaped, one hydrogel at a time. Every great discovery in science is a story of curiosity and seeing problems in new ways. The fight against cancer is one of humanity's greatest challenges, and its ability to adapt keeps us searching for new strategies. Imagine rewinding a cancer cell to its most vulnerable state. Not science fiction, but the cutting edge of biology. This journey starts with a simple hydrogel, a soft engineered material that mimics the body's natural environment. By placing cancer cells in this gel, scientists can coax them to behave like their ancestors. Stem cells. The goal: uncover the hidden switches that make cancer cells so dangerous. Traditional hard plastic dishes don't reflect the real 3D world cells live in. Hydrogels do. Changing a cell's environment changes its behavior, revealing vulnerabilities we can target. In this tiny universe within a gel, we can watch cancer cells shed their aggressive identity and revert to a primitive state. Each discovery brings us closer to smarter, more effective therapies. Creating the hydrogel is like following a precise recipe. The main ingredient is alginate, a seaweed-derived polymer dissolved in water. Special molecules and light link the alginate chains, forming a soft, stable gel. Its softness is key. Scientists tested gels of varying stiffness, discovering a Goldilocks zone that encourages cancer cells to reprogram. To make the gel a true home, they coated it with proteins that help cells attach, mimicking the body's own signals. Without these proteins, cells can't sense their environment or begin reprogramming. The team created thousands of tiny, uniform hydrogel droplets, each a miniature world for a few cancer cells. This innovation ensures every cell experiences identical conditions, making experiments reliable. Consistency is vital. Only then can scientists trust that changes are due to the gel itself. This careful engineering sets the stage for the next breakthrough. With the hydrogel ready, scientists introduced various human cancer cells, breast, lung, and bone. The question: would these cells revert to a stem cell-like state? Cancer stem cells are rare but drive tumor growth and resistance. To track transformation, researchers engineered cells to glow green when a stem cell gene, OCT4, switched on. Placed on soft hydrogels, some cells began to glow, proof of reprogramming. On hard plastic, almost no glow appeared. The green light was a simple, powerful signal. The gel's softness was triggering a fundamental change. This visual evidence confirmed the hydrogel's unique effect. Now, scientists could study these elusive cancer stem cells in ways never before possible. Observing glowing cells in a dish is one thing, proving their power in living organisms is another. Scientists injected reprogrammed glowing cells into mice. Even a tiny number of these cells rapidly formed tumors, unlike non-glowing or plastic-grown cells. This showed the hydrogel didn't just make cells glow, it gave them true cancer stem cell abilities. For the first time, researchers could reliably produce and study these potent cells. This breakthrough opened new doors, understanding what makes cancer so persistent. Now, scientists could search for drugs that target the very root of cancer's resilience. The hydrogel became a powerful tool for cancer research. With a way to create cancer stem cells, scientists could finally ask, what makes these cells tick? Deep genetic analysis revealed thousands of genes switched on or off during reprogramming. This genetic map pointed to key pathways keeping cancer stem cells alive. Using the Hydrogel system, researchers screened hundreds of drugs, watching for the green glow to disappear. Several drugs forced the cells to lose their stem cell properties and tumor-forming ability. One drug, targeting cell metabolism, reversed the reprogramming entirely. This journey, from a simple gel to a new cancer therapy, shows the power of creative science. By changing a cell's world, we may finally change the story of cancer itself. Science isn't just about big discoveries, it's about the careful process behind them. Like building a sturdy tower, science relies on honesty and transparency at every step. Scientists share their methods and results so others can check, repeat, and build on their work. This openness is crucial, especially in fields like cancer research, where lives are at stake. Strict reporting rules ensure every detail: how patients are chosen, what treatments are given, how results are measured, is recorded and shared. Without these rules, mistakes or misleading results could have serious consequences. So, when you hear about a breakthrough, remember, it's the result of careful planning, teamwork, and a commitment to transparency. This process separates real science from guesswork. It builds a strong foundation for future discoveries and better health for everyone. Science is a process we can trust because it's designed to check itself. That's what makes scientific knowledge reliable. Every experiment starts with a detailed plan, a protocol. This blueprint covers everything: how many samples to use, what tools to choose, and how to avoid bias. Sample size matters, too few, and results might be random. Scientists use statistics to get it right. Controls and randomization ensure fair comparisons and prevent bias. Every tool must be validated and calibrated, just like checking your measuring tape before building. Scientists decide on statistical tests before starting so they can't cherry-pick results later. Laying out all these details in advance keeps the experiment honest and reliable. This rigorous planning is the first step to trustworthy science. It's how we make sure results are meaningful, not just lucky guesses. Good science starts with a solid plan. After an experiment, scientists face mountains of data, far too much for any person to analyze alone. Powerful software helps organize, visualize, and find patterns in this data. In fields like genomics, computers piece together billions of DNA fragments, turning chaos into insight. Sharing raw data is key. Scientists upload it to public databases so anyone can check or reanalyze it. This open data culture lets others verify findings or even discover something new. If mistakes are made, they're more likely to be caught and corrected. Transparency in data keeps science honest and self-correcting. It's a global effort to make sure results are solid. Open data builds trust and drives progress. That's how science moves forward, together. The right materials are essential for good science. Scientists must validate cell lines and antibodies to ensure they're using the correct tools, like checking ingredients before baking. Validation steps like DNA fingerprinting confirm identities and prevent errors. Reporting these checks lets others trust the results. When animals are involved, strict ethical rules protect their welfare. Committees review every animal study to ensure it's necessary, humane, and follows the 3 R's: replacement, reduction, and refinement. Animals must be cared for properly: clean housing, good food, and veterinary oversight. These rules aren't optional. They're essential for responsible research. Ethical oversight and material validation are the backbone of trustworthy science. They ensure research is both accurate and humane. The ultimate test of science is reproducibility. Can others repeat your experiment and get the same result? Detailed reporting is essential. Every step, ingredient and condition must be shared. If others can't reproduce a result, it raises important questions and helps science correct itself. This process of repetition and verification is how science builds strong, reliable knowledge. When results are confirmed, our confidence grows. When not, we investigate further. This back and forth keeps science honest and prevents shaky conclusions. Precise reporting builds trust among scientists and with the public. When you hear about a new discovery, you can trust it's been rigorously tested and checked. Science's commitment to accuracy and transparency makes it a powerful tool for progress. Strong reporting is the map that guides us forward. That's how science earns and keeps our trust.
Hello everyone, and welcome back to the For Science Salah Lotfy channel. I'm Professor Dr. Salah Lotfy from the Egyptian Atomic Energy Authority, and today I want to pause for a moment and shine a spotlight on the brilliant minds who make groundbreaking research possible. Behind every scientific advance, there's a team of passionate researchers, and I believe it's important to recognize their dedication. This time, I have the honor to thank an outstanding group of international scientists whose tireless efforts have inspired me and, I hope, all of you as well. Let me extend my deepest gratitude to Dr. Jun Suzuka, Dr. Masumi Tsuda, and Dr. Lei Wang from Hokkaido University in Japan. Their work in cancer pathology and soft matter science is truly remarkable. I also want to thank Dr. Shinji Kosaka, Dr. Shinya Kojima, Dr. Toshihide Ueno, and Dr. Hiroyuki Mano from the National Cancer Center Research Institute in Tokyo. Your research in cellular signaling pushes the boundaries of our understanding. A big thank you to Dr. Karen Kishida, Dr. Shingo Sembus, and Dr. Hirokazu Sugino, whose commitment to scientific excellence at Hokkaido University is inspiring. From Tsukuba, I appreciate Dr. Sachio Abaratani and Dr. Yoshihiro Omiya, whose contributions from the National Institute of Advanced Industrial Science and Technology are invaluable. Dr. Martin Frauenlob, Dr. Takayuki Kurakawa, and Dr. Kazunori Yasuda. Your expertise brings new perspectives to life science and medicine. Special thanks to Dr. Jian Ping Gong for leading innovation across multiple disciplines. And finally, heartfelt appreciation to Dr. Shinya Tanaka, the corresponding author and driving force behind this research. To everyone involved, your hard work, passion, and precision set the standard for scientific achievement. I salute you. And to my viewers, thank you for joining me on this journey. Your curiosity and support fuel our mission to explore and share science. Let me know in the comments what part of this research excited you most, or if you have questions for the team. Together, we celebrate not only new discoveries, but the spirit of collaboration that makes science possible. Thank you for watching, and thank you for being a vital part of this scientific community.